Our next talk will be a tag team effort from the Department of Food Science. And again, the beauty of our research capacity, literally from farm to fork, from the cow's gut to the human's gut and everything in between. So our next two speakers will be Gisele LaPointe, who's a professor and DFO research chair in the Department of Food Science, and Tara Wilson, a PhD student working with Gisele, to talk about microbial milk quality from the farm through to the dairy product.
Thanks, Stephen. I'm really glad to be here. We're really glad to be here to make it onto the roster of all these high-hitting JDS publishers. So good to be here. You can see here we've seen nutrition this morning. We've seen genetics. We're going to completely change up and go to microbes, microbial milk quality. And the thing is since we're focused on microbes, I know Dave Kelton is going to be disappointed that we won't be talking about milk foaming. So I've put up the picture of June here, and I encourage you to go see June's poster, visit June, and he can explain much better than I can all the problems about milk foaming and all the neat discoveries that he's made recently. So focus on microbes, and to do that, I'm going to hand it over to Tara Wilson, who is a PhD student in my team, and that's her main thing, is working on milk quality. Take it away, Tara.
Thank you, Gisele, and I would like to say it's just an honor that Gisele asked me to help her out today, because I feel, as most people in this room, you love to talk about your work, and when you love what you do, you never work a day in your life, and that's personally how I feel with what I do. So first off, when I say the words microbial milk quality, what's the first thing that kind of comes to everybody's mind, and the major impacts of microbial milk quality? If anyone wants to yell out anything. I know quite a few people in my lab could probably yell out a few things, but... Shelf life, okay. Safety. What about more of the farm environment? Things like bedding, anything else like that? So I'm here to introduce a little bit of a different topic with microbial milk quality, and that is biofilms on farms. And whenever we speak about biofilms on farms, we ask ourselves three big questions. Who is there, how many, and what do they do? This is something that I don't think I need to address to anyone in this room, because, sorry. Thank you. As most people are either producers or well versed into the dairy industry, but just to keep everybody on the same page, I'm showing just kind of a simple rendition of a milking system. So starting from the very beginning with our teat cups, moving through the claw, all the way through milking hoses, the pipeline, filter socks, receiver jar, and milk holding tank. And when we further start to break this down, we see different components that kind of make up a majority of two separate segments of the milking system. So looking at the first half of the milking system, we see a lot of plastic, rubber, and silicone. And with plastic, rubber, and silicone, they can be prone to degradation via thermal stress, just general use and aging, as well as contacts with milk fats. This breakdown can lead to microscopic pockets and cracks that allow for protein and microbes to evade cleaning and sanitation. And we get to see this a little bit more close up as well with an SEM. So you can see general cracks and different degradations within a silicone rubber hose, which was exposed to fluid over time. And this brings up one of our main questions that a lot of people in the industry ask, and that's how often do we change hoses, and better yet, how often should we? Whether we're following recommendations by manufacturers, which can be at how many times of milking, or on an annual or biannual, or whether or not we see cracking, which is what some people may be doing. Focusing on the later half of the milking system, we see a lot of components made of stainless steel. And stainless steel is great, it's chemically resistant, but it does begin to wear down over time, which allow microbes to adhere. More formally in literature, this is called microbial-induced corrosion, and it's caused by a cathodic depolarization, which can cause deviations in the stainless steel and allowing for pitting and cracks, which again provide great safe havens for microbes to kind of evade our cleaning and sanitation. So in saying that, we moved on and we kind of tested out all of these areas that I just presented to you guys there. And we took a look at the residual microbes in milking systems, and this is of 31 farms, and this was sampled back in 2021. So all of these farms were sampled both in the spring and in the winter. And we pooled them together in order to be able to see log CFU per swab. So for anyone who's not a microbiologist in the room, a lot of the things that we talk about are in log CFU. And just to give a little bit of a heads up, when you look at your milk quality parameters, you have your 121 individual bacterial cells, which that can be converted to around 50,000 CFU per ml of milk. If we further convert that into stuff that microbiologists like talking about, which is in logs, that's the equivalent of about 4.7 logs, so just kind of below 5 there. So you can see here within our milking system, quite a few of them actually end up above our 4.7, specifically speaking rubber and stainless steel, which are two kind of major components that we like to look at. We further went into a bigger sampling set, combining both Ontario and Quebec, and comparing both conventional and AMS milking systems. So taking a look at this, total aerobic counts were higher in Quebec than Ontario, whereas in the milking machine, there was a tendency for lower counts in Quebec, specifically in the short tube. So just looking over on this side of the graph. Biofilms were higher in most inaccessible spots, and in Ontario, the load was greater in summer, with an average of 5.52 log mean compared to 4.49 in the winter. And then even more so, in robotic milking systems, total aerobic counts were higher in Ontario than in Quebec, except for udder cleaning brushes. And then more on to our original topic, which was to look at on-farm dairy processors. We did a comparison between commercial and then as well on-farm dairy processors for both Quebec and Ontario. So overall, the presence of biofilms in on-farm processing barns were similar to that of commercial dairy farms. Bacteria were higher in Ontario on-farm processors, and the on-farm dairy producers in Quebec manufactures actually raw milk cheese, which requires more stringent control in milk quality, which may explain some of the differences between Ontario and Quebec for our on-farm producers. So moving into our next big question that we always ask about biofilms is, how do these microbes stick around? So most microbes are removed through routine cleaning and sanitization, but certain genera can form biofilms which are resistant to cleaning and sanitizers. And we do have our known biofilm formers such as Pseudomonas, which is a big player in fluid milk spoilage, as well as Staphylococcus, which are some mastitis pathogens. And this has actually been studied a lot around the world, specifically in France, where they have designed pilot milking machines to show the ability of genera such as Lactococcus, Catheria, Streptococcus, Anoxibacillus, and Acinetobacter to create resident microbiotas despite cleaning and sanitation. So just to catch everybody up on speed on how Pseudomonas and kind of some Staphylococcus species actually work, this is what's known as classic biofilm formation. So it's a 5-step process, which usually starts with a reversible attachment where microbes are just very weakly bound to a surface. It's a temporary dipole. It's just more or less by chance. And then as that kind of progresses in around 2 to 10 minutes, that's when it starts to become irreversible. And that's when microbes really become hard to remove off of a surface. That's when there needs to be a greater increase in shear force, either by your slug or whatever your cleaning is. And if they're left even longer than that, that's when microcolony formation, mature biofilms, and dispersal really begins. And you might be saying here, looking at this timeline, going, well, that's not me. I do cleaning and sanitation. And my milk line definitely gets used before the 24-hour mark, so it's really not my concern. But this is where there's a proposed biofilm formation specifically for milking systems. And this is where we can see our routine cleaning and halogen-based sanitizers don't actually get to affect microcolony formation. Instead, they lead to an early dispersal, which can potentially attach further down the system. So you might see a problem that begins in the cup or the claw, which can potentially move down to the pipeline, which then can potentially move into raw milk. And it's actually really neat because now we have things that we're able to see some of these biofilms in working order. So this is a liquid dairy fill line, and it was actually identified by UV fluorescent. with a company that we've worked with before. And so they are there. It really comes down to what is in these biofilms and do they have a detrimental effect. So we decided to look at that a little bit further. And with that, we did a 6-month study of an on-farm processor. So here we swabbed both farm and facility, so 13 different swabbing areas, the same ones that I described before in the earlier image of the farm, and then an additional 19 swabs and 6 sponges of the facility. So this was an on-farm processor for us. All the swabs were taken after sanitation and in duplicate for both DNA culturing as well as culturable bacteria via plating. And one special thing about our producer was actually the ability for the milk was transported between farm and facility. So as much as it was an on-farm processor, there was still transportation of the milk. So looking first at our culture-dependent methods, we looked at 4 major media types. First, our M17 agar, which was sort of the equivalent of our total aerobic count. So any bacteria that liked mesophilic sort of room temperature, as well as used oxygen to grow. So that's a lot of your Lactococcus and Leuconostoc. We also did an MRS agar in anaerobic conditions for lactobacilli. S1 milk agar for proteobacteria, which is a lot of your environmental contaminants such as Enterobacteriaceae and Pseudomonas, and then finally a yeast glucose and chloramphenicol agar for our yeast and fungi. On the flip side of that, we also did culture-independent methods, where since everything was taken in duplicate, we actually got to look at the DNA communities of each swab. And for that, we chose to go with ASVs, so amplicon sequence variants, which are actual individual variants and individual genetic markers between bacterial types, which help us better identify. So sticking with the theme of ASVs, we looked at the shared and unique microbes between our farm and facility. So looking first at the Venn diagram A, you can see that the farm was more genetically diverse than the facility, but still shared 84 of those individual sequence variants between each other. We broke that down further into milk, water, and swabs, where you could begin to see that a lot of the ASVs between water and swabs were actually shared. Same with milk and swabs, and then there was a combined total of 44 that were shared among all 3 groups. This again was broken down to see what was shared just in the farm samples as well as just in the facility samples. So with both farm and facility, both shared ASVs between water and swabs, but then shared none between milk and swabs or milk and water. So when we look at things like ASVs, it actually allows us to take snapshots and to look exactly at what bacterial families and genre are impacting and are found in each. So looking first at our milk microbiota, in general, our milk remained under our 50,000 CFU ml for the duration of the study. So this was clean milk. This was not milk that had high BSN scores for the entire 6 months, and we went in there and said, okay, well, you've got bad milk, so we're going to see what's in it. This was clean milk. So looking at our culture independent, where we actually get to identify what bacteria are found in there, we can see that Lactobacilliaceae was more abundant in facility milk compared to farm milk, with that little transportation between farm and facility. Whereas Micrococcaceae and Brevibacteriaceae, which are both actinobacteria, remain the same between both milks. And Pseudomonas, which is our good fluid milk storage, biofilm former, remained low throughout the duration of the study. Next, we looked at environmental swabs from the farm and facility, so beginning first with our culture dependent. Our proximal milking system, which was comprised of our claw and hoses, remained above our LOQ, which is for us 1,000 CFU, all but 2 times over the 6-month period. But there was only 3 instances of viable counts exceeding the limit of quantification on food contact surfaces within the facility. So you can see on this side of the graph, this is all of our milking system, and then on that side is our facility. But for culture independent, only 51 swabs out of our total 356 that we collected contained adequate DNA for amplification on farm. In total, only 16 samples obtained from the facility contained adequate DNA, which was approximately around 5%. So in this, we can see a snapshot within this 5% of samples for sanitation in the facility. So only 3 of these were food contact surfaces that were collected before sanitation, so this was not an after sanitation practice. And these samples are mainly from sponges of the walls and floors. So there was a greater relative abundance of Pseudomonas and Lactococcus on sanitized surfaces, which does indicate potentially biofilms, but decreased Staphylococcus and Brevibacterium. But keep in mind, this is within our 5%. 95% of samples were clean and had no bacterial counts on them. So this is just relative to our 5%. From there, we can look at the transfer of microbes between the 2 facilities. So starting this way and going that way, just to guide you through a little bit. So starting over here with our farm and moving over to our facility. So this is done with ASVs, so the same thing that I was explaining before. These are individual sequence variants, so they can be attributed pretty closely to one singular thing. So we know if we see ASV 104 within the sponge, it is the same as it is within the drain. So for us, Pseudomonas remained low, and there was no counts between any of the farm. But Pseudomonas was found in high abundance within the sponge and the drain of the facility. But looking at Staphylococcus, we can see that it was low within the claw of the pipe of the farm, but then in high abundance within the pipeline of the farm, and then again seen in high abundance on food contact surfaces and brining within the facility. So we further went along with the Staphylococcus because we saw Staphylococcus, and we actually blasted these and made sure what they actually were. So this was Staphylococcus equorum for us, which, if anyone knows anything about cheese rind aging, that's actually a great addition to your cheese rind aging, as well as Brevibacterium down here, which showed similar patterns, being high in the pipeline and then again later in the food contact surfaces. So for us, this was great. It was beneficial transfer of microbes, which were fantastic for our cheese producer, who looks a lot for Brachybacterium, Staphylococcus equorum, as well as Brevibacterium. And these are actually samples taken off one of his cheese rinds to show that all of those microbes are there. So not always bad. Sometimes they're beneficial to cheese ripening color, but they aren't always. Unwanted fungi and bacteria can cause defects, specifically in cheese. A well-known one in washed rind cheeses is Scopulariopsis, which actually causes these black and dark kind of crevices creeping into cheese, which just makes kind of for an ugly cheese rind, and it doesn't make for very good sales. And then a more commonly known one is a spore-forming Clostridium, which, as most people know, can survive pasteurization. So what we then wanted to do after that was actually take a look at the cheese facility and what was kind of going on there, because our producer was struggling a little bit with a little bit of defects. So back to our facility, we ended up taking rind samples off different ages of cheeses. So we started at his 2-month cheese, a 5-month cheese, an 8-month cheese, and then a defective cheese, which was not going for sale. And of these, we took 20 scrapings off each rind, just so we were able to compare a little bit between our 2-month, our 5-month, our 8-month, and everything else like that. So you can see with our 2-month rind, there's predominantly Staphylococcus exilis and Penicillium. Moving into our 5-month good rind, we can see an increase in Staphylococcus equorum, the same thing that we were able to find in the farm, as well as still a good prevalence of Staphylococcus exilum, Penicillium, and Debromyces. And then that continues into our 8-month good cheese, where there's also a little bit more Brachybacterium, which really makes the good outside rind of your cheese. But when we start to compare bad month and good month 5-month cheese, we can see that we do get Scopulariopsis flava. And same with defective cheese, we do get quite a bit of Scopulariopsis fusca, which again promoted our defect within the cheese. So looking at this all in a big picture, we can say that microbes are everywhere in the farm, past production, everything else like that. And some biofilms can be helpful in aiding in the addition of beneficial microbes, but detrimental when containing pathogens or spoilage bacteria. And a deeper understanding of these interactions will help enhance cleaning and sanitation based on the species identified. And just to really bring this home with a conclusion, just so I don't get shot down by any of my funders saying, oh, you said we don't have to sanitize and biofilms are good for milk. No. So, on-farm processing facilities can contain microbes that can be beneficial to cheese ripening, but potential pathogens and spoilage microbes can also be present, and that's where we really start to see problems. So I'm not here advocating that we don't need sanitation and everything's hunky-dory. But a few recommendations to be made is, one of the big things that we can see is following regular maintenance and replacing of rubber silicone parts and gaskets, because that's kind of the first spot that you see bacterial accumulation in your milking system, can really make a big difference when moving to your end product. And the rapid elimination of milk residues, which we consider food for bacteria, as much as possible will help control biofilm development in milking systems, and best management practices are key to improving milk quality. So I'd like to thank everyone for listening, as well as my funders and Gisele for bringing me along on this ride, and I'd be open to answering any questions you may have.